Assalamu alaikum. I'm Dr. Sidra Arshad with the fourth lecture in the series of special senses. The topic is physiology of hearing, and I shall start by briefly describing the physics of sound waves. So sound waves, these are longitudinal waves. They are composed of region of compressions and region of rarefactions, then followed by regions of compression and so on. So when a tuning fork is made to vibrate, immediately next to the prongs, there is going to occur a region of compression, followed by a region of rarefaction, then another region of compression, and so on. So there are certain characteristics of sound which we have to know in order to see that how these characteristics are encoded by the auditory pathways and the auditory regions in the brain. So these characteristics are pitch, intensity, and timbre of the sound. So pitch refers to pitch or tone of the sound. It depends upon the frequency of the sound waves. Frequency means the number of sound waves per unit of time. So if a sound wave has a low note or a low frequency, it means that the number of waves per unit time will be less, as you can see in this figure. And if the sound wave has a high note or a high frequency, the number of waves per unit of time will be more. So in normal settings, the sound frequency is measured in units which are known as cycles per second, which are equal to hertz. So hertz means the number of cycles per second. So this is the frequency of the sound. You can see in this figure, even though these notes, they have different frequencies, however, they have same loudness. Same loudness is indicated by the same amplitude of the waves in both of these cases. So these waves, they have the same amplitude. That is why they are going to have same loudness. So the other property, which is known as loudness or intensity of the sound, it depends upon the amplitude of the sound wave. Soft sounds will have low amplitude, whereas loud sounds, they are going to have high amplitude. If we look at the relationship between the intensity and the tone, so low frequency sounds are usually louder, whereas the high frequency sounds, they are perceived to be softer in nature. There is another quality which is known as the timbre or quality of the sound. It depends upon the overtones. Overtones refers to the other sound frequencies which are present along with the fundamental frequency. So there is a pure tone, and superimposed on that will be sounds of waves of other frequencies. So together they impart richness to a sound, which is referred to the timbre or quality of that sound. So the audible frequency for the human ear is between 20 to 20,000 hertz. The greatest sensitivity is between 1,000 to 4,000 hertz. Male voice is usually of low frequency, whereas the female voice, it has a higher pitch. Male voice is about 120 hertz and female voice is approximately of 250 hertz. Let's look at the physiological anatomy of the ear. So the ear is divided into three parts. There is an outer ear, middle ear, and the inner ear. The receptors for hearing, they are found in the inner ear. So let's look at the outer ear. The outer ear is composed of this outer ear structures, which is known as the pinna. There is a central external acoustic meatus, which leads into the auditory canal. This auditory canal is separated from the middle ear by this tympanic membrane. Attached with the tympanic membrane are the system of three small bones. Immediately attached to the tympanic membrane is the bone which is known as malleus. Attached with the malleus is another bone which is known as incus. And attached to the incus or articulating with the incus is another bone which is known as stapes. Stapes in turn covers, or its faceplate covers, a window which is known as the oval window. This oval window, it leads to the inner ear structures. So inner ear structure for the hearing, we have an apparatus which is known as cochlea. Cochlea is composed of bony labyrinth along with the membranous labyrinth. In the membranous labyrinth lies the receptor for hearing, which is known as organ of Corti. Let's look at it in a little bit of detail. So the middle ear, it is characterized by these three bones, out of which malleus is closely associated with the tympanic membrane, inner respect of the tympanic membrane. Incus, it articulates with the malleus, and stapes is the bone which articulates with the incus, and it also covers the oval window. In association with oval window, there is cochlea. The cochlea is a coiled structure. If we uncoil it, it is composed of this membranous labyrinth, which is divided into these three compartments. So this membranous labyrinth is divided into three compartments. Immediately in front of the oval window is compartment which is known as scala vestibuli. Then there is another compartment immediately beneath the scala vestibuli. It is known as the scala media. The scala media, it houses the organ of Corti. Beneath the scala media is another compartment which is known as the scala tympani. All of these compartments, they are filled with specialized fluids. The fluids which occupy the scala vestibuli and scala tympani, they are known as perilymph, whereas endolymph is the fluid which is found in the scala media. You can see over here at the helicotrema, the scala vestibuli and scala tympani, they are communicating. There is another view of this inner ear structures, scala vestibuli, scala media, and scala tympani. So these compartments, they are separated from each other by these membranes. Between the scala vestibuli and scala media is a membrane which is known as vestibular membrane. Between the scala media and the scala tympani is the membrane which is known as basilar membrane. Basilar membrane is the membrane on which the organ of Corti lies. This right over here is the organ of Corti. So scala media, it is rich in the endolymph. It is backed by endolymph. It is also known as cochlear duct. Closely associated with the organ of Corti is the eighth cranial nerve, the auditory division of the eighth cranial nerve. This eighth cranial nerve, auditory division of this nerve, it innervates the hair cells. The hair cells are the receptor cells of the organ of Corti, and these are the cells which respond to the signals generated by the vibration of basilar membrane in response to the sound waves. Transmission through the ear. So the sound waves, they have to be transmitted into the inner ear. What happens is that when the sound waves, they reach the tympanic membrane, they strike it and they become vibrations. Attached to the inner aspect of the tympanic membrane is the handle of malleus, which is the first bone in the ossicular chain. These vibrations are then transmitted to these ossicles, making these bones to vibrate as well. These vibrations will then be transmitted to the oval window as the faceplate of the stapes. It is covering the oval window, which has a membrane over here. So this membrane is then vibrating and it is going to create fluid waves within the cochlea. So these fluid waves, these are generated in the perilymph. You can see over here these wriggly arrows. These fluid waves, they can push down on the flexible membranes of the cochlear duct. This right over here in blue, this is the cochlear duct which contains the endolymph. These fluid waves, they push down on these membranes, they set up fluid waves in the endolymph as well. These fluid waves, well, they are going to cause the vibrations of the ductorial membrane as well. As a result of that, the hair cells, they are going to bend, and this bending is going to open up certain mechanically gated channels, which we are going to see subsequently, creating the electrical signals within them, causing the release of neurotransmitters, and then this signal is then going to be transmitted as nerve impulses in the auditory division of the vestibulocochlear nerve. So you can see these waves, these fluid waves, these are pushing down on these membranes. When they push down on these membranes, they make the endolymph, they set up fluid waves in the endolymph as well. And then these waves, then they move to the perilymph of the tympanic duct. And from here, they move towards the round window, which also opens in the middle ear. And This way, the energy is dissipated. So there are two pathways which the fluid waves can take. They can push down on these membranes and cause the receptor potential to be generated in the hair cells, or these waves, they can move in the vestibular duct all the way at the moving to the tympanic duct at the helicotrema and then moving to the round windows. These waves, they can take these two pathways. So when we talk about the transmission of sound waves to the inner ear, the sound waves, they can take these three pathways. These are known as air conduction, ossicular conduction, and bone conduction. Air conduction simply refers to the transmission of sound waves to the fluid of the inner ear by not involving this ossicular chain. If this ossicular chain is not involved, the sound waves, they are simply going to cause the vibrations of the tympanic membrane, and these vibrations are then directly transmitted to the oval window, and from here on, the sound waves, they can, fluid waves, they can push on these membranes, or they can travel all the way to the round window through these compartments, that is the vestibular duct and the tympanic duct, or they can push down on these membranes and cause the activation of the hair cells. This is known as air conduction, which is not involving the ossicular chain. However, the ossicular chain is usually involved, and this is known as ossicular conduction, and this transmission we had just discussed. Bone conduction refers to the direct transmission of sound waves into the inner ear by making the bones vibrate. It is not going to involve the external ear or the middle ear. So what happens is that when a tuning fork, it is set to vibrate and it is placed on the skull, for example, anywhere on the skull, it is going to cause the vibration of the skull bones as well. So these vibrations will then be directly transmitted into the inner ear, setting up the fluid waves in the inner ear fluids and then causing the action potentials to be generated in the auditory nerve fibers. Matching is an important concept because the sound waves which are traveling in the air, they have to be transmitted into the fluids of the inner ear. Because the fluid, it has a higher inertia than the air itself, it is going to require an increased amount of energy for it to be displaced in order to set up vibrations into it. Luckily, there exists a system which acts as a lever. That system is malleus, incus, and stapes, and it is going to act as a lever. And by acting as a lever, it is going to increase the movement of stapes 1.3 times. Along with this, there is a difference in surface area of tympanic membrane and the face plate of stapes. This difference is about 17-fold. Together with the ossicular system and the 17-fold difference, it is going to account for 22 times increased force with which this fluid is displaced. So the force which was applied to the tympanic membrane, this force is increased about 22 times with which this fluid is now displaced and the vibrations are set up in it. This impedance matching, it is about 50 to 75% perfect for sound frequencies which are between 300 to 3000 hertz. By this impedance matching, most of the energy present in the incoming sound waves, it is preserved. It is seen that if the tympanic membrane is removed along with this ossicular system, the sensitivity for hearing it is going to decrease about 15 to 20 decibels. So this impedance matching is important to conserve all of the energy of the sound waves. There is another concept which is known as attenuation reflex. It is a protective reflex which helps protect the hair cells from being exposed to low frequency, high amplitude sounds. So what happens is that in response to loud sounds, the tensor tympani and stapedius muscles, which are present in association with this ossicular system, they contract. By contracting, they make this ossicular system rigid. This rigid ossicular system, it is not going to vibrate with as much amplitude as it does normally. So by this mechanism, the transmission of sound waves is going to be reduced. So it attenuates the low frequency sound transmission by 30 to 40 decibels. So significance of this attenuation reflex, it lies that it protects the cochlea from damage caused by loud vibrations, caused by loud sounds. It also helps mask the low frequency sounds in noisy environments. So if there is a lot of background noise and it is of high amplitude and low frequency, these sounds will not be transmitted as the ossicular system will become rigid. It also decreases hearing sensitivity to one's own speech. So this is the significance of the attenuation reflex. So the organ of Corti is the sensory organ for hearing. It lies on the basilar membrane in the scala media. This basilar membrane is in turn composed of a large number of basilar fibers. So on this basilar membrane are found hair cells, outer hair cells, and inner hair cells. There are about 3 to 4 rows of outer hair cells, while the inner hair cells are present in a single row. So outer hair cells, they are more numerous than the inner hair cells. These inner hair cells, these are the main receptor cells for hearing. Almost 90 to 95% of the cochlear nerve fibers, they innervate the inner hair cells, while outer hair cells, they are sparsely innervated. These hair cells, they are special in a way that their surfaces, they have stereocilia. These stereocilia, they are found in close association with a membrane which is known as tectorial membrane. These hair cells are also supported by other cells, testicular cells, and rod cells. So the inner hair cells, they are less numerous, however, they are maximally innervated and they act as receptor cells. Outer hair cells, they are more numerous and they are stimulated in response to loud sounds, for example, but they are minimally innervated and they control the response of inner hair cells. These outer hair cells, they also receive signals back from the auditory areas which regulate their depolarization or hyperpolarization, and by that mechanisms, they either they become elongated or they shorten, and then they control the response of inner hair cells. They are involved in the amplification of sound, frequency selectivity, and they also protect against the noise-induced hearing loss. So these are the functions of the inner hair cells and outer hair cells in the organ of Corti. Signal transduction in the hair cells. So whenever the tectorial membrane is displaced, it is going to cause the displacement of the stereocilia as well. So normally the hair cells, they are always firing at lower rates. They are firing, generating low rates of action potentials. What happens when they are stimulated? They are going to increase the rate of action potentials. So how it is brought about? When these stereocilia, they are displaced, they are going to be displaced in the direction of the highest stereocilium. So when they are displaced in the direction of the highest stereocilium, there is going to be opening of the mechanically gated potassium channels. As you can see, or at the top of these stereocilia, there are specialized potassium channels which are mechanically gated. That is, they are going to respond to the mechanical stimulus. And this mechanical stimulus is the movement or bending of the stereocilia towards the highest stereocilium. These channels, they are linked together by tip links. These tip links, they stretch and they open channels when stereocilia, they bend towards the tallest member. These channels, they open up and potassium, it moves inside of the cells. As you must remember that these stereocilia, they are exposed to the endolymph. Now, endolymph is a fluid which is secreted by the stria vascularis and it is rich in potassium ions. So these potassium ions, they move into the cell and the cell becomes depolarized. This depolarization, it opens up the voltage-gated calcium channels and the calcium entry is going to occur. This calcium entry, then it is going to cause the movement of these neurotransmitter vesicles towards the cell membrane, and the neurotransmitter is going to be released. The neurotransmitter over here is the glutamate. The glutamate, it is released and it binds to its receptors on the auditory nerve, and the action potentials are then generated in this auditory nerve fiber. Now let's look at the other scenario, which is the bending of stereocilia away from the tallest member of the group. If this occurs, the tip links, they slacken, and the channels, potassium channels, the mechanically gated potassium channels, they are closed. So if no potassium enters the hair cells, the cell hyperpolarizes, and this hyperpolarization, it is not going to cause the opening of the voltage gated channels, which are going to remain closed. These voltage gated channels are then closed and no neurotransmitter is released. When no neurotransmitter is released, there are no action potentials in the auditory nerve fibers. On the other hand, when the low rate of action potentials are being fired in the efferent fibers, that is the auditory nerve fibers, what happens is that there is lesser amount of opening of these mechanically gated potassium channels. They are open up to some amount, some potassium can enter. However, the cell is not hyperpolarized. So a few calcium, voltage gated calcium channels will open up, and the vesicles, they will move towards the cell membrane of this hair receptor, and it will be released, causing low rate of action potentials in the afferent fibers. So these three scenarios, they exist. However, if the tectorial membrane is displaced, and this displacement also causes the movement of stereocilia towards the highest member, then these mechanically gated potassium channels are going to open up, and potassium is going to move inside of the cell, bringing about depolarization and eventual release of neurotransmitter, and action potentials will be generated in the auditory nerve fibers. So when the auditory nerve fibers are stimulated, they are going to carry the impulses all the way to the auditory cortex. So let's trace the auditory pathway. So the auditory nerve fibers from the spiral ganglion of corti, they are going to synapse in the dorsal and ventral cochlear nuclei, which are found in the upper part of the medulla. Here, all of the fibers will synapse, and the second order neurons which will arise, they are going to either cross over to the opposite side or remain as such, but they are going to travel to the superior olivary nuclei, where they are going to synapse as well. Two pathways are going to diverge from here. Some of these fibers, they are going to travel as lateral lemnisci all the way to the nucleus of the lateral lemniscus, while most of the fibers, they are going to synapse in the inferior colliculi. The fibers which had moved to the nucleus of lateral lemnisci, most of them will move to the inferior colliculi, where they will synapse, while some of them will synapse with the medial geniculate nuclei. Well, all of the fibers which had synapsed in the inferior colliculi, they are then going to synapse in the medial geniculate nucleus. This medial geniculate nucleus is the nucleus of thalamus. From here, the fibers, this is the relay station, and from here, the fibers, they will be moving to the all the way to the auditory cortex. So multiple crossing overs, there are multiple levels in which these auditory fibers, they are crossing over to the opposite sides. They include the trapezoid body, the commissure between the two nuclei of the lateral lemnisci, and in the commissure connecting the two inferior colliculi. So it means that if the left organ of corti is stimulated, the right auditory cortex along with the left auditory cortex is going to perceive that sensation of hearing. Also, the collaterals, they move to the reticular activating system along with the cerebellar vermis. So let's look at the characteristics of the auditory cortex. So the auditory cortex, it is found in the superior temporal gyrus in close association with the sylvian fissure. There are two auditory areas which are important: auditory cortex area 1, a primary auditory cortex, and association areas. The primary auditory cortex, also known as the Brodmann area 41 and 42, and then there are multiple association areas, out of which the area 22, Wernicke's area, is quite important. So what are the functions of primary auditory cortex? The primary auditory cortex is involved in the tonotopic organization. In the primary auditory cortex, there exist tonotopic maps. That is, the different sound frequencies are perceived at different areas of the primary auditory cortex. As you can see over here in this diagram, that anteriorly, the low frequency sounds, these are encoded, they will be perceived, and high frequency sounds, they are located posteriorly. So the tonotopic maps, they retain the spatial organization of the frequencies which are perceived by the hair cells, which depend upon the movement or the displacement or the vibration of basilar membrane along different points. And this is retained in the auditory cortex as well in the form of these tonotopic maps. Different frequencies of sound are processed in specific spatial locations within the cortex. This area is also involved in discriminating sound pitches and pattern of sounds, perception of pitch, loudness, and direction of sound. Secondary auditory cortex, these association areas, they integrate auditory information with other stimuli, for example, the visual stimuli. They are involved in the higher level auditory processing, that is speech comprehension, music perception, and the recognition of environmental sounds. They are also involved in the language processing. For example, the vernix area, which is the important area for language comprehension. So it is seen if the primary auditory cortex is removed, it is not going to completely diminish the animal's hearing. However, it is going to greatly reduce or abolish the ability to discriminate the different sound pitches and the pattern of sounds. And if the secondary auditory cortex is removed, a person is not going to be able to interpret the meaning of the sound which is heard. So these areas of the auditory cortex, these are quite important. Now let's look at the functions of some other structures in the auditory pathway. The superior olivary nuclei, they are involved in sound localization and the processing of binaural two-eared cues. They basically localize the sound, that this sound was heard either in the left ear more or the right ear more. So basically, they are involved in the localization of the direction of sound. Especially the medial nucleus, it does that by measuring the time lag between the acoustic signals reaching the two ears, whereas the lateral nucleus of the superior olivary nuclei or the lateral superior olivary nuclei, they interpret the difference in intensity of the sound reaching the two ears. For example, if the intensity perceived in the left superior olivary nuclei is more, then it means that the source of the sound was closer to the left ear. Inferior colliculus is involved in the integration and processing of auditory information, including sound localization, pitch, and intensity. So you can see that these features, they are being distinguished at the lower areas, that this discrimination between sound localization, pitch, and intensity, it does not occur in the auditory cortex alone. It begins in these lower areas. Whereas the medial geniculate nuclei, they act as relay stations, sending the processed auditory information to the auditory cortex. So how the frequency of sound is determined and then encoded? There are two mechanisms. The number one mechanism is known as the place principle. It states that the discrimination of frequency or determination of frequency can be made by seeing the place of maximum stimulation of hair cells along the basilar membrane. So if we stretch out the basilar membrane, we can see that the area of the basilar membrane, which is close to the oval window, it is maximally stimulated or maximally vibrated by the sounds which have high frequencies. So high frequency sounds, they are going to maximally vibrate the basilar membrane, which lies close to the oval window. So high frequency sounds, they are going to maximally stimulate the hair cells which are located near the base of the cochlea. So the area which is close to the oval window is also known as the base of the cochlea. Whereas the low frequency sounds, these low frequency sounds, they are going to maximally stimulate the cause the movement of basilar membrane near the apex of the cochlea. The apex of the cochlea is close to the helicotrema. It is away from the oval window, quite away, very far away from the oval window. And you can see that the sound frequencies which lie between the high and low, that is the intermediate sound frequencies, then they are going to cause the maximal stimulation of hair cells along these intermediate areas, the areas between the two extremes, that near the oval window and that far away from it, close to the helicotrema or apex of the cochlea. So why does this difference occur? As I had told earlier, that the basilar membrane is composed of these basilar fibers, and these basilar fibers, they have different characteristics at different places. So the basilar fibers which are close to the base, close to the oval windows, these are short and stiff, and these fibers, they become long and limber near the helicotrema or the apex of the cochlea. So these fibers, they are varying in their stiffness and their length. That is why they are going to respond to different frequencies. The lengths of the basilar fibers, they are increasing progressively, beginning at the oval window and then going from the base of the cochlea to the apex. So the place principle helps in determination of frequencies between the range of 200 to 20,000 hertz. For low frequency sounds, that is between 20 to 2000 hertz, and other principle is involved as well, which is known as the Pauli or frequency principle. It is involved in the perception of low frequency sounds. What happens is that when low frequency sounds are encountered, the groups of neurons, they work together in a coordinated manner, firing in a volley or alternating pattern. So these group of neurons, they are going to respond to these low frequencies and they are going to fire together in a coordinated manner. This firing in a coordinated manner is going to be perceived as a frequency by the different areas in the auditory pathway and the auditory cortex. So this collective firing, it allows the brain to encode and perceive the frequency information of the sounds, which exceeds the temporal limitations of individual neurons. So that is why they are now working together and firing together. So in this way, lower frequency sounds are normally detected. The characteristic of the sound is the loudness, which is measured in decibels. So to determine the loudness of sound, there are certain principles, mechanisms in place. Loudness of sound is determined by the amplitude of vibration of basilar membrane. That is, louder the sound, greater the amplitude of vibration of basilar membrane. So hair cells which will be excited, they are going to discharge, they are going to get stimulated and send action potentials down the nerve endings at more rapid rates. So if more hair cells will be involved, there is going to be spatial summation. That is, many nerve fibers instead of only a few fibers will be involved because more hair cells are now getting excited. The loud sounds are also stimulate the outer hair cells. The outer hair cells, they are not stimulated in response to soft sounds. However, they are stimulated in response to loud sounds, and when these outer hair cells are stimulated, they are going to apprise the nervous system about the loudness of sound. So the signals which are generated in the auditory nerve fibers, which had innervated the outer hair cells, they are going to reach the auditory cortex, for example, and they are going to give the information that the sound is loud. So the direction of the sound is determined by two processes. One is that the time lag between the entry of sound into one ear and its entry into the opposite ear, it is perceived by different structures along with the auditory cortex. And this works well for sounds which have frequencies less than 3000 hertz. For higher frequencies than this, the difference between the intensities of the sounds in the two ears can help with the determination of direction of the sound. That is, if the intensity of the sound is perceived as high in the left ear, it means that the source of the sound is closer to the left ear. So the intensity of the sounds are determined at different levels of the auditory pathway and at the level of auditory cortex as well. It can also help with the determination of direction of sound. So now let's look at the abnormalities of hearing. The loss of hearing is known as deafness, and it is divided into two categories depending upon the cause. So conductive deafness refers to impaired sound transmission in the external or middle ear. Conductive deafness means that the transmission of sound waves, the conduction of or the transmission of sound waves to the inner ear is somehow, it is obstructed, it is impaired. So the impaired sound transmission, it occurs at the level of external or middle ear. This type of deafness, it is for all sound frequencies. Some of the causes include accumulation of wax in the external canal, for example, or any foreign body getting lodged over there, and also the infection or inflammation of the external acoustic meatus, which is known as otitis externa. Infections of the middle ear or otitis media, recurrent pharyngitis, especially, it can predispose, especially children, to otitis media, which is the infection of the middle ear because of the communication through the eustachian tube. So this otitis media is going to cause the buildup of fluid in the middle ear, and this buildup of the fluid, it can also lead to the perforation of tympanic membrane. Another disease which is known as otosclerosis, it causes the replacement of the bones in the middle ear with the fibrotic tissue, and it can actually cause the sclerosis of the faceplate of stapes, and this is going to then cause the decreased vibration, decreased ossicular conduction, and this is going to cause conductive deafness. On the other hand, sensory neural deafness, as the name indicates, the problem lies either at the level of receptor or the auditory pathway, anywhere in the auditory pathway. So it could be because of loss of cochlear hair cells. The loss of cochlear hair cells, it occurs with age and with exposure to loud sound especially, or if there is damage to eighth cranial nerve, the cochlear division of eighth cranial nerve within central auditory pathways, or this type of sensory neural deafness, it does not involve all the sound frequencies. This deafness occurs to certain pitches. It can also be seen in Meniere's disease, in which there is buildup of endolymph fluid, which can damage the organ of Corti. It can be seen in presbycusis, which is the age-related loss of cochlear hair cells. Then there are certain antibiotics, aminoglycoside antibiotics, especially streptomycin and gentamicin, which interfere with the functioning of the hair cells. Prolonged exposure to loud noise, certain autoimmune disorders, traumatic injuries, certain tumors which involve the auditory division of the vestibulocochlear nerve, or even the vestibular tumors compressing the auditory nerve, or seen in the case of medullary stroke. So anywhere in the auditory pathway. Even though a thorough history, it can point towards the cause of the hearing loss, but there are certain tests which have to be done in order to differentiate between the type of hearing loss, that is either it is conductive or sensory neural, or even mixed. So tuning fork tests, they are used to differentiate between the conductive and sensory neural types of hearing loss. However, they are useful only when one ear is affected, If both the ears are affected, these tests are not much useful. And they also do not quantify the hearing loss. That is, these tests, they do not tell us that how much of the hearing loss is actually there in terms of decibels. So these tuning fork tests, they can be done in the clinical settings, either in the OPD or on the bedside of the patient. They involve making the tuning fork vibrate and then using this tuning fork to assess the hearing of the patient. So these three tests are used: Weber, Rene, and Schwabach. The method of Weber's test is that this tuning fork is set to vibrate, and then the base of vibrating tuning fork is placed on the vertex of the skull, and the subject or patient is asked whether he perceives the sound of this tuning fork equally in both ears, or if he is hearing that sound more so in one ear than the other. The normal result is that the person, he or she hears the sound equally on both sides. In conductive deafness of one ear, the sound is going to be louder in diseased ear. Now, what happens in conductive deafness when the normal ossicular and air conductive pathway is obstructed? The bone conduction, it is actually going to become better. It becomes better because the masking effect of environmental noise, it is absent on the disease side because the sound waves, now they are not passing through the external ear into the middle ear, where the attenuation reflex was one of the mechanisms by which the sound was masked, environmental noise was masked. So the bone conduction, it becomes better than the air conduction in a diseased ear, which is involved in the conductive hearing loss. On the other hand, in sensorineural deafness, obviously the sound will be louder in the normal ear because that normal ear has the normal organ of Corti and the normal auditory pathway. So sound is going to be louder in the normal ear. So this has to be remembered that in conduction or conductive deafness, what happens is that the bone conduction, it becomes even better than air conduction because air conduction is now actually compromised because of the obstruction. In Rene's test, a base of vibrating tuning fork is placed on the mastoid process, it's present behind the ear, and the subject is asked to indicate when he no longer hears the vibrations of the tuning fork. Immediately when the subject indicates that he is now not able to hear the vibrations of the tuning fork, the prongs of the tuning forks, they are immediately placed in front of the ear, that is in front of the ear canal. And if the subject is able to hear these vibrations, it means that his air conduction is better than bone conduction, which is a normal finding. That is, the person, he hears the vibration in air even after bone conduction is over. This means that there is no hearing loss. This finding is known paradoxically as Rene's positive. So Rene's positive test is actually normal, in which air conduction is better than bone conduction. However, in the case of conduction deafness, the vibrations in air, they are not heard after bone conduction is over. The bone conduction was better than air conduction in conduction deafness, so the person will not be able to hear the vibrations of the tuning fork when it is held in the air next to the ear. That is, the person is not going to be able to hear that in the affected ear in which there would be conductive deafness. So this is known as negative Rene's. In sensorineural deafness, the vibration heard in air after bone conduction is over as long as nerve deafness is partial. Person will be able to hear some sound when the tuning fork will be held next to the ear after the bone conduction is over. So this is going to indicate that there is sensorineural deafness. The tuning fork test is the Schwabach test. In this, bone conduction of patient is compared with that of the healthy subject or with the examiner himself, if his hearing is not impaired. In this, a tuning fork is set to vibrate, and then it is passed back and forth between the subject and the examiner. Both of them should stop hearing through the bone conduction at the same time. If they do, then they are normal. However, if the subject is able to hear for a longer time through bone conduction, it means that his bone conduction is better than normal or that it is lengthened. This will indicate conduction deafness. If the patient cannot hear for a long time, he hears it for even a shorter time than the examiner, then it means that his bone conduction is even worse than normal. That means that he is not actually able to perceive the sound. The problem must be at the level of the sensory receptor or in the auditory pathway, which indicates sensory neural type of hearing loss. So these are the tuning fork tests which can be used to differentiate between conductive deafness and sensory neural deafness. However, when there is mixed type of hearing loss present, the tuning fork tests, they are not of much help. Whenever the hearing loss has to be quantified, a specialized test known as audiometry is performed. In this test, an individual is seated in a soundproof room and the audiometer is connected to him via headphones. Sounds of various frequencies are then presented to the individual's ear through these headphones, and the individual has to indicate when he hears that sound of the particular frequency at which intensity level. These recordings are then plotted on a graph, and this graph is known as the audiogram. So the audiograms can be used to differentiate between the types of hearing loss. They can indicate which type of hearing loss is present, either it is sensory neural, conductive, or mixed type of hearing loss. And they also tell us that how much of the hearing loss is actually there. So let's look at these two audiograms in front of us. The first audiogram, it indicates that this is the normal hearing level, and these markings or these plots indicate the hearing level of the subject being tested. So the crosses represent air conduction, whereas the stars, they represent bone conduction. So you can see that the hearing level of the individual, it is not normal, it is actually lower than normal. So it indicates that there is hearing loss. Y-axis indicates the loss in decibels. So hearing loss is present over here, but you can see the plots for air and bone conduction, they are close to each other. So it means that both the air conduction and bone conduction are approximately affected equally, and this indicates sensory neural type of hearing loss. And the loss for the sounds which are of higher frequencies is more so as compared to the loss which is occurring at the lower frequencies. And we had already discussed that in sensory neural deafness, there is loss of hearing for some pitches more so than the other pitches. So this is the sensory neural deafness. Audiogram for the conductive deafness, you can see over here, the stars, they indicate bone conduction, and the crosses, they indicate air conduction. There is a gap between these two. This is showing that the loss for air conduction is more as compared to the loss for bone conduction. And this is the classical finding of conductive deafness. There is loss for both the air conduction and the bone conduction. However, the air conduction is affected more than the bone conduction. So the audiograms can help us quantify the hearing loss, and they can also be used to diagnose conductive and sensory neural types of deafness. Auditory evoked potentials are electrical recordings which can be obtained from the surface of the body. That is, electrodes are placed on the scalp, behind the ears, and even at the earlobes. And these electrodes are going to record the electrical activity when the sound is being transmitted in the auditory pathway. So this is an objective assessment of hearing and auditory processing. It is going to provide valuable information about the function and integrity of the auditory system. So the aberrant recordings anywhere in the area of the auditory pathway, if there is any problem, they can indicate at which level the problem actually lies. They can help guide clinical decision-making in the management of hearing disorders. For example, they are used to assess hearing loss, screen newborns and young children, especially who may not be responding correctly to the subjective test, can be used to diagnose auditory pathway disorders and monitor auditory function, especially those who have been prescribed hearing aids. These are the study resources which were used to prepare these lecture slides. You can study from these places on your own. Thank you so much. If you have any questions, please contact us on any of our social media handles.